good morning, everyone. Thank you so much for being here this early in the morning. Hope everybody got their caffeine ready. So in the next 30 minutes, I'm going to talk a little bit of scientific events that we have at Alloy Therapeutics. Hopefully that will goes well with your coffee, and then let's get started. So today's talk, we'll talk about two arms that we have at Alloy's for antibody discovery. First of all is our immune discovery branch, and then the second part will be on antibody optimization. Starting from the first part is our rapid on-cell selection with yeast display. For whoever is familiar with antibody discovery, I'm sure we all have our challenges that we met in our career, and one of the most challenging part is actually find the native antigen format for our selection. And a lot of time for membrane protein specifically, we always handle with misfolded protein when we express it solubly or for the extracellular domain. And especially when we're dealing with a structure with flexible loop, it makes things even harder. And then for the second part, we also experience low extracellular domain region in general. Take GPCR for an example. There's less than 10% of the overall GPCR math is expressed outside of the cell membrane, and that makes the targeting of that specific receptor very difficult. And the third part is the protein purity. I would say as of nowadays, still soluble protein is one of the most popular or direct forward antigen for our selection. But a lot of time when we purchase it from commercial vendors, we often see there might be some cases showing low purity, and that would significantly impact our selection process. And also, in terms of expression, we obviously will have a protein tag linked with our antigen for purification purpose, but that tag sometime would also interfere with our selection and lead to failed campaign process. And what does those challenge mean in actual practice? Well, the first one is obviously we'll have limited diversity. And second, it will actually give us poor recovery. And then we would also have loss of function clones that we can ultimately sort it out from the whole process. And at Alloy, we actually tackle this problem step by step. And first, we have our very nice proprietary humanized mouse strains that we are utilizing for our immunization. And that fundamentally helps us to get a really diverse pool to begin with. And for those difficult targets, we are actually using mRNA LNP strategies for our immunization to ensure that we actually have a very large repertoire to begin with. And that granted us always have really nice, robust titer, and then in the end, retrieve high-affinity binders. And second stage is our antibody recovery stage, and here is where we introduce the yeast display platform. And we often will just take the immune tissues out of our Alloy mice and then put that into our yeast display platform. And I know every platform has its own advantage and disadvantage, but in terms of the diversity, yeast display platform is definitely offering us way larger repertoire or diversity compared to B cells or hybridoma platform. And one thing that I do wanted to emphasize today is that along with the yeast display, we also develop on-cell binding sorting strategy that would essentially allow us to sort against native expressing antigens on the cell surface in this very native format. And that would ensure us to find out the binder that ultimately is the one that's functioning and is what we wanted in the end for therapeutics. And the last part is our high throughput characterization process at Alloy. After we finished all the sorting and enrichment process, we would go ahead and do our NGS and then selected clones using our in silico IIML platform to select out both a diverse pool of the sequence and of course also the enriched sequences that we have discovered through yeast display. And we will go through high throughput protein production called MapForge process. And here we can produce hundreds to thousands of clones within a couple of weeks, and then we will go through a high throughput characterization process that would again take another couple of weeks. So within the matter of 1 month, would be able to characterize in depth of up to 1,000 clones from what we have discovered from yeast display. And next, I just wanted to kind of go through a couple of case studies with you guys that we have recently done at Alloy. The first one, of course, is GPCR. It's everybody's favorite target. So in this case, I just wanted to emphasize again how important that we were able to sort it on cell-based target, because GPCR in general is very hard to express solubilely. I know there's a lot of technology have evolved to try to express GPCR outside of membrane and make it solubilized, but then the structure of it is still hard to solve and then maintain its native format. So in this particular case, we leveraged our advantage in terms of on-cell sorting. And then for this particular project, we were told to select against a GPCR that has to have cross-reactivity with its cyno counterpart. And then the format of the antibody that we're dealing with is an SCFV. And we went through 5 rounds of selection, and then in the end, we were able to retrieve most of the antibodies that with single-digit nanomolar EC50 on cells, and then some of them is even sub-nanomolar. And in the middle of the panel, you will see that's how we did our directed evolution. On the x-axis is the extent of how yeast library bind to the either GPCR expressing cells or the knockout cells, which is our negative control. And then on the y-axis is the rounds of sorts that we have done, and then we can see a very clean background on the knockout cells throughout from round 0 to round 5, and then we have really nice enrichment going from round 1 towards round 5, that binding to the target GPCR on the cell surface. And then we also did single clone characterizations and showing at the bottom, with the blue curve showing is your target GPCR binding, and the red curve shows is your negative control. And then for the case that's showing on the right, you will see that our target binder is binding to specifically to the GPCR on the cell surface and not binding to our negative control. And then towards the right, you will see this is what I was talking about, the high throughput cell binding characterization. We're able to characterize thousands of clones in one set of assay, and we define our binding with different colored of lines. So basically anything between red, yellow, and green is what you wanted to have negative, medium, and positive control. And what the box defined with the the box defined with the green lines is the box that you wanted to have, meaning that your clones are actually binding to your target without binding to your negative control. And as shown in the graph, the majority of the clones that we have tested actually bind to very specifically to the human GPCR target that we have tested. And overall, we screened 190 clones, and then the vast majority of it binds to the human target. And then a very decent percentage of it binds to the cyno target, and 19 out of 24 of them have no polyspecificity flags for the developability downstream characterization. And then the case study 2 I want to talk about is TCRs. It's another hard target that recently have emerged as a very interesting target of people look for antibodies targeting. And in this case, we're actually lucky in the sense that we do have soluble protein of the TCR available, but then we would also wanted to validate the binding on cells at the very end of our selection to make sure that whatever antibody that we have discovered throughout the process is actually binding to the TCR on the T cells and would be able to be functional for downstream process. So in this case, we displayed Fab on yeast cells, and then we went through 6 rounds of selection. And then in the end of the day, we were able to retrieve a very diverse pool of binders with single to double-digit nanomolar KD. And then in the middle, again, is showing the flow plots of how we're doing the sort. For the x-axis, we're always showing the expression of the various constructs expressed on the yeast surface. And then for the y-axis, we were showing either the binding to the TCR protein or the TCR expressing cells. For the top row, we're just showing that for very simple and common strategy that we use the TCR protein as our bait and then sort out the population that bind to our TCR protein. And then second line is showing when we're confirming out of the selection, does the population that we have sorted actually bind to the cells? So what you can see on the left is our negative control, and we can see on the top right quadrant there, you don't see any binding showing that whatever selection that we have done is actually very specific, and the binder doesn't bind to the cell itself. But then going on to the right graph, you see that very nice enrichment boxed out in the red outline there, showing that whatever population that we selected previously actually binds very specifically to the TCR on the cells. So after we have had the enrichment, we again went through our high throughput characterization process and confirmed that for sure we did have TCR specific binders. And again, after the vigorous selection, we were able to retrieve very diverse pool of binders that we can provide to our partners for any drug therapeutic downstream process. And the third case, which is the last case for this part, I would like to share a little bit of our cross-reactive VHH discovery. So in this case, it's a really interesting story because in this case, our partner wanted us to develop VHH against both human and cyno version of the target. And we had no problem validating the human target on Octet. The way we do our QC is basically load our human protein or cyno protein on Octet and then flow it with comparator antibody that is known to bind with both target. And we validated, as you can see in the middle panel, that the human protein bind to the comparator antibody without any problem. But then the cyno protein doesn't bind to the comparator at all. So that was a huge red flag at the beginning of the project, and we were like immediately recognized, whoops, we can't actually use cyno protein to do our selections. So we pivot in the middle of the project after we sorted against the human protein. We utilized the cyno expressing cells that would help us to finalize and narrow down the binders that is both human and cyno cross-reactive. So again, at the bottom of the middle panel, you will see on the left is before you sort, there is very minimal amount of enrichment in the top right quadrant. And then after we sorted against the cyno positive cell lines, we have a very nice enrichment towards the cyno target binding. And then we did our high throughput characterization, confirmed that the binders that we were able to pull out from the selection are both human and cyno cross-reactive. And the attrition rate in this particular case is slightly higher than what you have seen in the previous two cases. Largely was due to the homology of the cyno target is actually slightly less than what you will see in general compared to the human format of the protein for the previous two cases. So that wraps up the first part of this talk, and hopefully I convinced you that at Alloy we were able to do on-cell rapid sorting, and that would allow us to deliver targets that would kind of in generally conventionally considered as hard target, but then we were able to deliver a very diverse pool of binders with high functionality. Next up, I want to share a little bit instead of the discovery front, I want to share a little bit more about the affinity maturation. I'm sure everybody here have heard enough of affinity maturation talk for the previous 2 days of the conference. And here I just wanted to hone in into yeast display strategy specifically. And as we all know, there's a couple challenges for antibody optimization as well. One being that, or the driving force of any sort of antibody optimization is affinity maturation. And the reason for that often comes from the number of copies of targets that are expressed on the target cells. Take TCRM as an example, right? A lot of time when you're having TCRM targeting a new antigen that is presenting on the MHC, we're talking about in general less than 50 copies of the targets on the cell surface. And if you compare a very common and traditional oncology targets such as HER2 or EGFR, we're talking about more than 1 million copies of target on the cell surface. So we're dealing with a very day and night type of situation in terms of developing a specific TCRM. So we are definitely required to develop sub-nanomolar affinity binders that would give us the efficiency that we're looking for. And secondly, it's also hard to maintain your specificity while you're doing your optimization. For example, when we're talking about TCRM, it would only specifically to that peptide MHC because it specifically recognizes a couple of residues on the peptide. And if you're really focusing on your affinity maturation, it's very easy that you lose the contact of those key residues through mutating the CDR sequences. And therefore, you lose your specificity targeting your TCRs. And then lastly is that it's very easily to introduce any sort of polyspecificity or reactivity throughout this very, I guess, brutal force, forcing it to have a high affinity. And that would further introduce any sort of developability flags downstream for the drug development process. So at Alloy, we again tackle those problems stage by stage. The first stage of our process is always to profile the sequence of interest. Oftentimes, our partner would come with us with their favorite clones or their favorite sequence in mind, and what we will do is do a thorough study of the targets. What we wanted to figure out is within that sequence, with the environment of the framework of that wild type already have, what does each amino acid contributes to in the CDR region specifically towards the antigen binding and its developability. So we will introduce single point mutations in each CDR region, and we have all 6 CDRs included. And then we'll introduce those mutations into each display platform, and then we'll sort based on the product profile criteria. And that would allow us essentially draw a profile chart for our specific wild type sequence. Then will tell us what amino acid at what position will give us beneficial or not beneficial property towards our end goal. And then once we have that profile, we'll look deeper into the sequence itself. And here is just an example of the data that we will get typically at the end of this process. The top graph that you will see have all the blocks lightened up is often our run 0 library before we go into any sort of specific sort fitting to the product criteria. And on the y-axis, you will have all possible 20 amino acid available. And then on the x-axis is at each position of the CDR loops. And here you're just seeing 3 CDRs from either heavy or light chain. And for run 0, you want to have all the boxes lightened up, meaning that we hit most majority possibility of amino acid switch in each position. And then going to the middle graph is when we're looking at specifically the non-binding population during the sort. It is important because that information would teach us at what location or region of the CDRs that would render this entire clone from a really good binder to a non-binder. And that's definitely the region that we would want to avoid. And then the bottom part is the region that we sequenced and looked at what will give us an improved binding affinity. And for this particular case that is very interesting, we have a very nice separation between our binding and non-binding cases. And then it seems like the top part, which is this CDR1 region, give us the benefits specifically for binding affinity improvement. And then CDR2 and 3 are the regions that render the whole antibody into a non-binder, are the ones that we wanted to be careful when we do antibody engineering. So after we have all this information, obviously we can annotate further of the structure, and we say, okay, maybe the CDR1 is the part that we wanted to put in more thoughts and then engineer for affinity maturation, and then CDR3 is the part that we actually want to stay away from, and that would most likely introduce any non-binder or polyspecificity in that region. And then after that, we will combine all the point mutations together into a combinatorial library, and then again put that into our East display platform to do downstream selection. So finally, after we have sorted our enriched clones that carries the desirable functionality and the characteristics, not just the high affinity, but also in terms of any, let's say, pH sensitivity or competition targeting each specific epitope, we'll put those through our high throughput characterization workflow again. And here we were in depth characterizing the kinetic functionalities and then also the cell binding of the binders. And then we also look at in-depth of the developabilities throughout all different kind of aspects that I listed on the slide. And then finally, after we have done all of this, I wanted to show you a couple successful cases that we went through recently for TCRM optimization. For the two projects that I'm showing here, each of them are required to have affinity matured to either lower than 100 picomolar or 200 picomolar, which is pretty high standard. And then in each case, we have from 2 to 3 wild type to do the optimization from. And then from the case from the left, you can see at most we can achieve 340-fold affinity improvement while maintaining its target specificity and then maintain the developability of the construct. And then for the case showing on the right, for all antibody, we were able to achieve more than 100-fold affinity improvement, and at most we were achieving more than 640-fold affinity maturation. And then one case from the project 1 is also very interesting that we see from one of the wild type sequence. To begin with, it actually is not the perfect clone because it shows a little bit of polyspecificity to begin with. What I show here on the flow plot is our first characterization on the wild type on the yeast cells, and then we utilize the PSR region incubating with the yeast cells and to see whether it would non-specifically binding to it. And we do see a slight upshift in terms of the binding signal, kind of indicate that yes, we need to be careful if for the normal cases we were optimizing trying to avoid adding any sort of developability flags. In this case, we're actively trying to remove any existing developability flags. So when we're doing the yeast cell sort, instead of only looking at the affinity, we also look at its binding to the PSR region, and then we kind of look at our NGS data and try to do the cross comparison and see at what position does it not lighten up to binding to the PSR region, but it does enrich towards the affinity maturation part. So that is the amino acid and region that we're heavily focusing our engineering. And then to grant our final design that when we tested for the developability, what you see is the wild type showing the darker color and then the variants showing slightly light blue color. We did achieve our affinity maturation goal, but at the same time also improve the developability property of this clone. So hopefully at this point, I have shown you our capability of doing affinity maturation and also beyond with our yeast display platform. And we can carry out the entire process from the generation of variants till the end, very detailed characterizations, again, within 12 weeks. And then we can carry out this optimization process with multiple variants at the same time. And then we have routinely done up to 3 antibody optimizations in parallel within the same time frame. So as a conclusion, I hope I have convinced you in the past 25 minutes or so that we were able to discover specific clones that is targeting native formats expressing on the cell surface. And with our Pol4 yeast display platform in-house, we're able to carry out antibody optimization that tailors specifically to partners' needs. And thank you so much for your attention, and again, really appreciate everybody show up super early in the morning, and I would love to take some questions if you have any.